Welcome everyone to week number 5 in Science 200. We are still working on our projects. We've got one extra little bit to add to it. And then after next week, we will get to making your presentation. So let's see what we've got going on. So last week, in week 4, we talked about scientific communication and how there are different ways to communicate science. And you have to decide who your audience is in order to tailor your presentation or your information to that particular audience, because you need to know what they already know, what they already understand, what their viewpoints are, what their opinions are on the issues, so that you can tailor your message to them, but also anticipate any questions that may come up so that you will have the answers ready for them when they do ask those questions. So scientific communication is terribly important in the world today. More scientists need to learn how to do this, because you should be able to explain your research in a matter of a few minutes to anyone who asks you what you do. You ought to be able to boil it down to just a few sentences to explain to someone what it is that you do and what you've discovered so far. So this week is all about Chapter 5. You've got more work to do in your Sumo web text, and you're going to see in the work this week in the textbook how important it was for scientists to become involved in the Deepwater Horizon oil spill, because the BP scientists were saying that only a certain amount of oil was coming up out of the ocean floor, and the scientists determined that no, it was basically 10 times that amount on any given day. And so different strategies had to be developed in order to stop the flow of oil. Now you're thinking, well, why was there such a difference? Because the BP scientists wanted to keep this quiet. They wanted to minimize the effect of this out there in the general public and what the general public thought. BP certainly wanted this, but what they were telling everyone was not the truth. And so we had to get to the truth through the science. So you will see that in your web text this week. We will also look at biases in science and how important something like a double-blind study is. A double-blind study is where neither the researcher nor the subject, client, patient knows what type of treatment is being given. And this way, there's no bias. There's no researcher bias. There's no confirmation bias. There's no subject bias in the study. You're trying to take out bias as much as you possibly can, and there's all types of biases that we try to minimize through our scientific studies. So you're going to learn more about biases. You're going to learn about p-values. So normally when you're asking a question in science and you're looking to see how probable this thing is that is happening, you say, okay, let's just assume that there's no difference between the two treatment groups, right? Maybe you're investigating the use of a new type of drug in people who have some type of illness, and you just assume that there's not going to be any difference between the two groups. If your p-value that you calculate is 0.05 or less, what you say is that no, there's very low probability that there is no effect. So that's why you're looking for a p-value of 0.05. So that means 5%. There's a 5% probability or less that there is no difference in the treatment and the control group. So you're saying it really is happening. You're looking for an effect. Then on the other hand, you have confidence levels, and that's sort of the opposite of p-values, if you want to think of it that way. So you're looking at confidence levels of 95% or greater that this thing actually could be happening. You think this thing is happening. It falls within the range of data that you have and that you've looked at. So those are confidence levels. Why do we use these terms? Because we want to know what is the probability that this thing is actually being caused by what you're saying it's being caused by. You're looking at all the data that's out there. You're looking at all the data that you've collected to check to see if this is a statistically significant event that has been happening. That's why you're looking at p-values or confidence levels. And along with that, in the textbook, they give you the example of Gregor Mendel and his pea plants and his studies in genetics. So back when he was growing his pea plants, people did not even understand that DNA was genetic information. They might have known that there was this molecule called deoxyribonucleic acid that was in cells because they could chemically analyze it, but they had no idea that that was the hereditary information in cells. So no one understood that. And they didn't understand how traits were even inherited. Gregor Mendel came up with the ideas of dominant recessive traits. He came up with the idea that each individual pea plant has 2 copies of each of the traits present, and that sometimes that trait is hidden, and so he called it a recessive trait. And sometimes, if it was present, it was always showing. That was a dominant trait. The reason that Gregor Mendel could figure all of this out was because he did a tremendous number of experiments with pea plants and examined all different characteristics from flower color to plant height to color of the leaves and the stems and etc., etc. He looked at all different traits in these pea plants because there were lots of different types of pea plants that farmers planted. So he studied them and he analyzed the data using statistical analysis. And he figured out that yes, there had to be 2 copies of each trait in each of these pea plants. And that's how you could get dominant and recessive traits. And that's how you could get recessive traits showing up in later generations that you didn't see for a couple of generations. So he explained this by counting. He had a tremendous amount of data that he collected. I think that he raised over 29,000 pea plants in his career as a geneticist. So he was a monk, and you know, monks don't have a lot to do except sit around and pray. So he was a gardener and he loved plants. And so he did these studies. Now, when he got promoted and he became an administrator, he quit growing his pea plants, and that was the end of his genetics, which is, in my book, hilarious. But anyway, that's just my commentary. So Gregor Mendel shows the importance of collecting data and analyzing the data correctly. All right, and then there's no discussion board postings this week. But you do need to revise your rough draft according to the feedback that I'm giving you on assignment 4. And you're going to add a section, and this is called the hypothesis section. So in the beginning, you came up with a question, right, to ask about, ocean acidification, electric vehicles. How carbon neutral really are electric vehicles, or how fast is the ocean becoming more acidic, or what is the effect of lowering the pH on marine life in the ocean, or goodness, what else? What's the effect of cutting down trees on biodiversity? Something like that. You came up with a huge question, right? A big, overarching kind of question. That's what you came up with in the beginning. All right, now you're going to come up with a hypothesis. A hypothesis is much more specific, and you should be able to devise some type of experimentation to answer your question. So it's got to be way more specific and down on a level where you could actually find an answer to it. So it might be, will the addition of a basic substance to ocean water affect the pH of it depending on how much you add, right? And you can figure out that you could devise an experiment around that. You could take, I don't know what just happened. You can take different ocean water samples and make them be different pHs and then add something like baking soda to each one to see if you can change the pH and how much you have to add in order to change the pH. So you can figure out how to design an experiment around that. So your hypothesis is a much more specific question that you can answer through experimentation. And so you're going to devise a hypothesis and figure out how you could answer this. Use your rubric and your guidelines to guide your work, and make sure you read my feedback, which is on this next slide. I'm just saying, use my feedback, because I'm telling you whether you're doing things correctly, whether you need to fix anything. If you need to fix something, I'll tell you. Usually I'll tell you how to fix it. And if you're not sure, if you don't understand what I've written, please, all you need to do is ask me. m.sigmon@snhu.edu. I'll be glad to explain it. I'll be glad to help you with it. If you can't think of a hypothesis, I guarantee you I can help you with that. Okay? So don't get stuck with whatever you're doing. Just ask me and I will be glad to help you. All right, with that being said, I hope that you have a wonderful week number 5.